motivational and very informative session. I learned a little bit now about vaccines and why they are so important and why it's a challenge to make pan-coronavirus vaccine. There are lots of questions. I will just ask one question, then I will hand it over to our expert. My question is, I still remember the day you called that this is going to last long. It's very severe kind of, right? I thought it was early days. And then I didn't realize, and obviously you're expert, it was true. We still are suffering from this pandemic. Thanks for sending us that informative video. You and Dr. Noman Abbat, who is also an immunologist, and that video was very informative through our outreach sessions. Thanks for being with JK Scientists and being a patron of JK Scientists. So I will not ask many questions, any questions even, because I don't feel I am even able to talk about it because I'm not expert. But our Sprout mentee, Umayya Muzaffar, who is a PhD student at Malaysia, and she's also coordinating many programs, mostly outreach programs at JK Scientists, will start a question and then she will take it forward. I think she has maybe one answer to your challenge. Maybe, I guess, you will enjoy her question as well. Thank you.
Sounds good. Thanks, Mubarak.
Thank you, Dr. Raiees, for such an enlightening and insightful talk. It's really an honor to have you here, and I'm really fascinated by the research that you're conducting. So especially the emergence of SARS-CoV-2 variants of concerns and the generation of neutralizing antibodies that could be really a cure for treating the COVID-19 crisis. So beginning with my question only, I have a question regarding the neutralizing antibodies. I have read somewhere that the high mutation rate may lead to the escape of the virus from neutralizing antibodies, even after a single mutation. So the neutralizing antibodies against SARS-CoV have limiting blocking effect on SARS-CoV-2 despite the high similarity in their RBD structures. So how can the neutralizing antibodies be enhanced with the new variants with high mutation rate?
Yeah, I think that's a very important point that you're raising. So you're kind of suggesting that once we generate these antibodies, they could lead to variants because they effectively put selection pressure on the virus where they bind and viruses could mutate. It's kind of definitely something that's considered, and it does happen. And most of these mutations around. So if you have those mutations that I showed at 417 or 484 position in RBD, since those are the most frequently elicited responses in humans, and those mutations recurrently happen in SARS too. So there is a suggestion that those could be driven by the antibody responses that you generate, and those lead to escape mutations. Generally speaking, I mean, obviously that's kind of like, that's kind of a potential issue, but it's mostly if you have antibody responses that are suboptimal. So if you have antibodies, for example, let's say you need 50 molecules of, well, we can talk in terms of molecules and the quality of molecules. You need 50 molecules of antibodies to effectively neutralize a virus, means completely clear it. But if you have only 10 copies of that antibody, it's a suboptimal response. So that in that scenario, the chances of escape could arise. And then it's also like, how effective are those molecules? Do they tightly bind to the virus? If they don't tightly bind, it leaves a scope that the virus can also escape. I think it's a very complex subject, whether or not, and it's kind of a highly discussed area on flu as well. Like, we get flu infection and vaccination literally every year we get vaccinated. And do those antibody responses drive antigenic shift or antigenic variation? I think there's a consensus that if the responses are suboptimal, there is chance of escape, be it the quality of antibody or the titer of antibody that you have in circulation. There is chance of escape. But if you have a highly effective response, there is little scope for the virus to escape. I think for SARS-CoV-2, there's a general suggestion that a lot of these mutations are generated in immunocompromised individuals, where you could have suboptimal responses, or you could have virus sitting in them for a long, long period of time because the body is not effective enough to clear the virus. So instead of having the virus for 5 or 10 days, you could have it for a year or maybe longer. And people have isolated viruses from some individuals after a year or like 6 months. So then if it stays longer, it has more chances to mutate. And in the immunocompromised individuals, the immune system works differently. The selection rules are kind of different, and how you select mutations, and these mutations are generated all the time, but then it's also like how they are selected. So I would say a general consensus is that current variants, especially Omicron type variants, are generated in some immunocompromised individuals rather than antibodies that are induced by vaccination or infection driving these mutations. So, but I agree, some of these mutations, if you have suboptimal responses, could be selected by vaccine responses.
It's really interesting. Thank you for clarifying. So the next question is from the participant, namely Raiees Andrabi. She's an MS from Ames. She is already here in the meeting. You can unmute yourself.
Ramesh, are you here? You get an opportunity to ask question directly to the expert. Maybe she has some network issues.
All right, I'll ask on her behalf. So she's asking, with the vaccine, aren't we allowing SARS-CoV-2 to evade immune system, which eventually leads to mutation?
It's kind of a similar question. So I think obviously, like there's a school of thought that thinks that it could happen. There's a potential. But I would emphasize again, if you have suboptimal responses, then there are chances for that leakiness. But if you have a very effective response, there's less chance to. But I don't want to rule it out. It is a possibility, and it could happen. And some of those mutations in the RB, those could be generated by vaccine-induced antibodies in humans.
All right, thank you. Another question from Obeda Akhtar. Her question is, how does STING pathway respond to the coronavirus?
I'm not super familiar about STING pathway, but generally speaking, I think coronaviruses can act. I think there's reports that. Sorry, there's some background. So I think if you can activate STING molecules, they are effective against coronaviruses. For example, OC43, which is an endemic coronavirus. So if you activate STING pathways independently, they can. They are generally like they have antiviral activities. So if you can activate those pathways, they are effective against viruses. But coronaviruses, if you infect cells with OC43, which is endemic beta coronavirus, they're not generally good at inducing STING pathways. But you could generate, and there is a suggestion that STING pathways might be able to have some role, and people are pursuing some therapeutic strategies against SARS-CoV-2 or other coronaviruses based on STING pathways. But there is not clear kind of understanding about whether or not in natural infection you can activate STING pathways. Maybe there are, but I'm not super familiar with that area.
All right. Perhaps one more question from Zafarullah Zargar, PhD faculty. His question is, Dr. Raiees has developed antibodies against RBD domain of spike protein. Is this antibody effective against new variants of COVID-19 virus like Delta variant and Omicron?
Yes, I showed that data in my slides, and as we would have predicted, because these antibodies that we isolated, they target more conserved epitopes, and those epitopes, we've done some structural studies also that I didn't show today, that show that these antibodies target regions that have not mutated in the Omicron variant, and many of those antibodies are effective against Omicron variant, and some of them are equally effective against Omicron as compared to the Wuhan strain. So yes, they are effective against Omicron, which is a great news. Because if we look at the field in general, so in this paper, we described around 40 broadly neutralizing antibodies, and we characterized around 30 in detail. And many of those neutralize SARS-CoV-2 effectively. But if you look at the rest of the field, most of the antibodies failed against Omicron, especially the antibodies in clinic. So this gives us more opportunities in terms of pursuing these antibodies for antibody-based prophylaxis or therapeutic interventions. And we are actively pursuing those options now because these are effective against Omicron.
That's interesting. Actually, there is still a lot of vaccine hesitancy, and regarding those hesitance, I have been reading a very good sentence that Omicron is a clarion for finding a pan-coronavirus vaccine, and it's a wake-up call to the world that the vaccine, how vaccines are so important. So in that view, there are two questions which are linked to the vaccine hesitancy. One is from Aisha Mushtaq. She's asking, clinical trials of COVID vaccines in children and its reliability. Tackling vaccine hesitancy and the infodemic at ground level. Is vaccination enough for now, or how long shall we expect to continue with different waves of variants?
Oh, that's actually many questions. So let me take those one at a time. Yeah, so yeah, I totally agree. I think it's obviously vaccines. We have seen with many pathogens, if you want to eliminate a disease, and I think it's going to be extremely hard to eliminate SARS now. It's likely going to be endemic, or almost certainly going to be endemic now. So it is extremely important to have intervention strategies, not just for the SARS-CoV-2, but for any potentially emerging coronaviruses that could cross in future. And I can almost certainly say that they will, especially the sarbecoviruses that use ACE2. So yeah, it's high time to make vaccines that have broad capacity to effectively neutralize these viruses. As for the clinical trials in children, they have shown a high efficacy, and obviously, and they've been shown to be safe. Obviously, you do the dose is less than what you would use in adults, but they have proven to be safe. And I think to completely curb this pandemic, we would need to vaccinate kids as well, because they could be carriers and they could be keeping, as long as we can contain this virus and reduce its spread, I think that's the best case scenario going forward. And what was the other question?
How long shall we expect to continue with different waves of variants?
I think it's very difficult to predict. I think I don't think there's a certain timeline. Unfortunately, single-stranded RNA viruses, they mutate, and coronavirus is not new to that. So we expect this will mutate. I guess it depends on so the virus has three important features that are important to monitor. One is transmissibility, and Omicron seems to be really effective at transmissions. It's highly effective. It's definitely more effective than Delta, which was kind of the previous crazy variant. And then escapability, and Omicron seems to be again really good at that. And then the pathogenicity, or how severe diseases it causes. So those three factors, viruses generally try to balance between the three, and it's actually remarkable, escaping so much from the antibody mediated immunity. Omicron is still so effective at transmissions, and hopefully I think there is some evidence that the disease that Omicron causes may be less severe. That might be a good news, but we have to wait for more data, because we have to specifically look at unvaccinated, uninfected people to see clear differences between other variants. But yeah, clearly there's it's difficult to draw a timeline when this is going to stop to generate variants. I think the best strategy is to contain it, take as many measures as possible, so that it's an experimental lab for this virus, the whole world right now. It as long as it keeps infecting people, it will keep changing. Which direction, I don't think we can predict.
You're right. The science is evolving, especially for SARS-CoV-2 research, and we need to be patient until the vaccine gets fostered in the future. So we have a question from a viewer on Facebook, Said H. Mir. He says, good to see you, Raiees. Hope you and Romana are doing great. A question that prizes my mind for a long time is, as per the book knowledge, a vaccine based on whole particle like Covaxin and Sinovac, etc., should be more effective, at least theoretically, as they present all possible epitopes on surface of virus to our immune system. Why they are for some reason not so commonly used world over? Is there any scientific reason or commercial interests are prevailing here?
Thank you, Said. And yeah, I think that's a very good point. I mean, that's kind of the classical way of vaccinology. Covaxin is made by chemical inactivation. So you chemically treat virus with a chemical, and you can inactivate it. And it was kind of a traditional way, and I agree that you could generate all sorts of responses. You could generate a T cell response, and you could generate a B cell response, and also you could generate responses to multiple parts of the pathogen, which is kind of how you generate natural immunity. I guess the issues are heterogeneity, how those virus-like particles are going to behave in different individuals, and how homogeneous preparations can you make, because one of the most important features for vaccines are consistency, how effectively they can, obviously how effective responses they can make, but also how consistently can you make those responses. So it's kind of important in that way. But I'm pretty sure, especially in India, the Covaxin has been pretty decent. Like, its efficacy has been pretty comparable to other pathogens. I think there is some hesitancy in the current age to use these classical ways of vaccines. Maybe that's partly why we don't make these vaccines that way, but it's still kind of an option, and a viable option. And obviously the heterogeneity issue is kind of a downside. So, and also I think there is some vaccine hesitancy, because even if you heat kill or chemically treat a virus and use that as kind of a whole virus vaccine, there's possibility that some of it might still be replication competent. And I think people hesitate to take those types of vaccines because there is an outside chance that there could be still viable copies of the virus that could cause infection. It's rare, but it could happen for the inactivated or killed vaccines.
Great. So the next question is from Mohammed Sharik. He says, recently, Professor Andrew Pollard, the director of the Oxford Vaccine Group and head of the UK's Committee on Vaccination and Immunization, gave a statement. We can't vaccinate the planet every 4 to 6 months. It's not sustainable or affordable. What are the chances that there will be a vaccine that overcomes the booster problem in the near future?
Yeah, I think that's a really, really important point. And I kind of agree with what was said, because I think there's a couple of issues here. One is obviously variants are arising, and even if you have an effective response to the wild type virus, the variant it may not be effective against the variant. So that's one problem. But then also there's the problem of waning immunity, and it's a general phenomenon for respiratory viruses. The immunity doesn't last long, especially coronaviruses. It starts to drop after some time, and we've seen that we get a really beautiful response early on, and by 4 months it's kind of still there, but by 6 months it starts to drop. It could be associated with the way coronavirus infects humans, or the way these vaccines are made. I think one of the major issues with coronavirus and respiratory viruses in general, and I kind of emphasized it, is how do we generate immunity at mucosal level, not just in the blood? Because if you have antibodies circulating in your blood, they can reduce the disease severity because the virus can't go in. And that's how these vaccines are working now. They're not necessarily preventing infection because the mucosal immunity wanes over time, but they are good at preventing hospitalization because you have very strong immunity in blood. So I think we have to develop ways how to train our immune system with vaccines to generate protection at mucosal levels, so that if the virus comes in, there is protection there. I think that's where kind of the major issue is right now with SARS. Even if you have effective immunity in your periphery, but if you don't have mucosal protection, you can still get infected. And you could still, the problem is that you could still be a carrier and transmit to other people, because the virus just proliferates in the upper respiratory tract, and it can be transmitted to more people without even causing a severe disease in that. You could be even asymptomatic, but it can still replicate. So I would say, yes, I think finding ways to develop strategies that could induce, especially the antibody responses that can last longer at mucosal surface would be key. And then also, as I kind of alluded to in my talk, targeting more conserved epitopes is kind of key. Even if then it addresses both issues of waning immunity as well as any potential variants of concern that may arise.
It's really interesting to know, as the questions are many, and the next parts will be taken over by Dr. Syed Mubarak Hussain. Thank you.
Thank you. This is so interesting.
Thank you, Maya.
It's been already 1.5 hours, and there are so many questions. I'm sorry we cannot take all, but I can club all of them together in 2 big questions, which I guess is very important for young students and young scientists to know the answer for. First, if I could group them all together, questions from Muntaha, Mubina, Namia, and Ikra, is what are the career opportunities in immunology and vaccinology? What is the secret of your success? What is your advice for a young researcher or young master, PhD, or even postdoc scientist? And how do you get admission at your institute on scholarship basis?
Okay, that's a lot of questions again. Okay, so the first part is how do we apply to different places for pursuing higher studies and research essentially. I think JK Scientists has a remarkable, they've done a remarkable job, and we have a lot of programs that kind of guide students through these processes for admissions and stuff. And most of the places in Europe and US, I think you could apply directly to the grad school programs, and they support your tuitions and stuff, and also your research work. So I would suggest like send as many applications as possible, but obviously like there's a lot of people like Said Mubarak and others who have done a tremendous amount of work over the years in guiding people and mentoring them, how to prepare your CV or like your application, which is extremely important. We probably don't pay much attention to that. And, obviously it's part of like, where we come from, and understandably, but, iterating those and seeking the right guidance and, right, asking for right mentorship is kind of key. And there's n number of opportunities. I don't think there's any lack of opportunities, just like actively pursuing them. And I think Mubarak will agree with me on this. And there's like a lot of opportunities. You just have to contact the right people and go prepare in the right direction and take the right steps. And then the next question was opportunities at Scripps. Yes, Scripps has a grad
immunology and vaccinology in general.
Yeah, so there are many institutes that are more focused on vaccines or immunology, Scripps being one of them. One of the biggest centers for HIV vaccine research is Scripps, mainly, thanks to Ian Wilson, Dennis Burton, and other people who have kind of led that field over the years. Duke also has a very strong program on vaccinology. Rockefeller, many other institutes. Harvard has many. And generally speaking, the grad programs, if you get into the grad programs, they support your education and also your research and other stuff. And as I said, I think there's very strong mentorship programs in JK Scientists. And if you are interested and are looking for any suggestions or guidance, they can suggest you. If you have any questions for me, you can email me directly, and I'll try to help as much as I can. But there, again, kind of emphasizing, there's plenty of opportunity that you could look for, and with the right kind of guidance, it shouldn't be that difficult.
What is the secret of your success from Ratnipura to Scripps? Define in 3 minutes, 2 minutes maybe.
Well, I think it's kind of a relative term, success. What do you call success? And I think you could be happy with your contributions that you make in any field, and enjoy your work as much as you can. I think that's what I would kind of see as a success. I think at the end of the day, if you are happy and if you're passionate about what you're doing, I think that's the major, the most important thing, and it drives you in the right direction. I think you have to ask yourself honestly, like, if you like something and if you are passionate about it, you can find ways to be successful in that area, but you have to be really passionate. And one of the things that I feel like that has helped me, and I'm sure many other people in different fields, is I think hard work is key. And there's, I don't want to like kind of be redundant and say there's no substitute to hard work. It's obvious. I think hard work is key, and being very honest about like what you do and how you execute things, and celebrating every moment in life. Like even if an ELISA plate lights up in my lab, it gives me happiness, and because I'm passionate about like, oh, this was my hypothesis, it's working in the right direction. Celebrating every bit of your journey is kind of key, and keeping the momentum and being very passionate and being very honest with what you do, because that gives you a lot of motivation if you're passionate about something. And yeah, that's all I have to say about that.
And also I would add to all the advice you have given is mentorship is very important, and collaborations, which I guess
absolutely I couldn't cover all of them, but yeah, absolutely.
But also
I think
training, right?
Yeah, no,
you
absolutely.
I think from somewhere.
Yeah, I think one of the things that I kind of, on a personal note, I realized was that mentoring is, part of like, if you think about method education, like, properly channeling your efforts and energy in the right direction. Like, what do I read? Like there's with internet, and, so much information online, you could easily get distracted, and you could be like, for example, if I think about vaccines, for example, there are so many pathogens. If I'm not focused on a few vaccines that my work is related to, I could be easily distracted. And so kind of, thinking about that constantly and crystallizing that in your brain, what you need for the next steps, is actually extremely critical. And it's that mental exercise is important because it shapes your brain in the right direction and you take the right steps. And, obviously mentoring and, other, there's so much information these days, and it's extremely important to be vigilant about like, what exactly do you need? And I would emphasize method education is extremely important, because as I'm growing older, I feel like doing things in a methodical way is extremely helpful. It does help. It makes you more effective. You can zoom out all the distractions and really focus. But if you go with a method, for example, as Mubarak highlighted, having a mentor or a guide could help you. It's just months of education. If you talk to some person who has first-hand experience, you don't have to go through the same, you don't have to reinvent the wheel. Like they will tell you exactly, but you have to also, it's also important. And I've seen a lot, I don't want to be complaining, but I've seen a lot, people don't have a lot of patience in listening to someone's experience. You have to listen carefully what they're saying, because they have learned it through a process. And if you listen to them carefully and kind of execute your plans in this in the right direction, you will be successful. And if you do it in a proper method, through a proper method, it will reduce the amount of time that will take you from A to Z.
Thank you. So one piece of advice for PhD students.
Just pick your topic carefully, like what you want to do, because this is something that you're going to be doing for next 4 or 5 years. You need to be passionate about something that you want to do. It's okay if you delay your research work by 6 months or a year, if you are not getting the right lab at the right time. I would say you do some other things, that will build your experience in the direction, but choose a program a topic that you are really passionate about, because it's really difficult to keep to stay passionate, or it requires a lot of patience, just to kind of, say it simply, it requires a lot of patience in PhD. There's exciting moments where it's mostly a lot of patience investment. So pick the right topic, things that you're really passionate about, so that you know that you can do it. And it's PhD is almost like a committed step in a biochemical pathway. Once you're committed in this direction, you will be doing it for the rest of your life. So you just be careful about picking the right topic for your PhD, and right place also.
And one piece of advice for postdocs.
I don't think postdocs need my advice.
Right.
They're already committed. Yeah, again, I think passion is like extremely important for science. I don't think you can go for long if you're not passionate about what you do. I mean, that goes for any field, but I would say like science tests you more, your patience more. So better kind of like, choose something that you think you can do for the rest of your life. Even if it's, related to, like, you could go in different directions. You could become an independent scientist. You could become a teacher who teaches science, or, something related to that. You could join a company, but it's still like you're doing the thing that you chose. So I would emphasize that, picking something that's that makes you interested and passionate about the subject is kind of key. From my personal experience.
Sorry, thanks for your suggestion and advice. One last question. There are lots, but should we all take vaccines? Does it drop fertility rate? Should we take booster dose? Is it worthy? Have you taken COVID-19 vaccine?
It's not one question, it's multiple questions, by the way.
Together.
So yes, I think the booster is great. If you get an opportunity, just go for it, because there is enough evidence now that if you are 3x vaccinated, like if you have gotten 3 shots, there's more protection against Omicron, which is going to be the most dominant variant going forward for some time. So there's more protection as compared to two vaccine shots. So definitely science behind getting a booster shot if you get a chance. I think India has started vaccinations for probably immunocompromised and more than age 60. But as soon as you get a chance, definitely. I don't think, I don't know if the protection is going to last for long, but hopefully for at least 4 to 6 months. But then, riding these waves is kind of like a containment strategy right now. Like, as long as you want to prevent yourself from getting infected, I think that's kind of that should be the priority, regardless of whether it's going to give you a lifelong or like long-lasting protection. I would say as soon as you get an opportunity, just get vaccinated, the 3rd shot. And people who haven't gotten vaccinated, they should definitely, I would really encourage them to get vaccinated, because even if it doesn't prevent infection from Omicron, it does prevent hospitalization and other disease complications. We don't know the consequences of, like, if you get infected with SARS-CoV-2, because there's this long COVID thing that you could have virus in your body for a long time. It could infect it could stay in your brain for long, and people are experiencing fatigue and other severe symptoms, and you don't want to be in that situation. Although with Omicron now, I feel like it's extremely difficult to avoid getting infected because it's so infectious. But you could still try with proper social distancing, masking, and taking the right measures, and getting boosted at the right time. So I would encourage, as a vaccine also, I would encourage, if you get a chance, get boosted.
Thank you very much. Thanks. I will hand it over to Umaya and we conclude the session. Thank you.
Thank you,
Dr. Mubarak. There is one person, Anuj, waiting for his turn. You can unmute yourself.
This is the last question then. Sorry.
Yeah. Am I audible?
Yes, I can hear you.
Yeah. So first of all, thank you so much. It's a quite informative session. Sir, actually, I have also have two questions that small questions that, sir, how is it possible to have vaccine for SARS-CoV-2 in such a short span of time? Do it need to be stability with respect to time, or we are just having this at very early stage? And second question is that why there is no vaccine for HIV till now?
So your first question, could you please repeat it one more time? So you
Yeah.
said you,
How is it
if I
possible
understood
to
it,
have the, yeah, how is it possible to have a vaccine for SARS-CoV-2 in such a short span of time? Do we need more stability like that with respect to time, or we are just having this at a faster rate, or what we are expecting
I
from
see.
this over the long period?
I see. I think, as I kind of emphasized in my conclusion slide, I think the biggest advantage we had, obviously we had some knowledge about coronaviruses based on research that was done at Vaccine Research Center, NIH, and other labs in US and across the globe. We had a lot of information already based on MERS and SARS-1. mRNA technology has been the major force in this rapid response to SARS-CoV-2. I don't think if we didn't have mRNA technology, and that's kind of what has helped to respond to this pandemic so quickly, because if you think about other conventional approaches like making a protein-based vaccine. So for example, if you have a subunit-based vaccine, you have to first develop a cell line that would secrete that vaccine, and it takes like I've done personally that for an HIV vaccine. It took like a year and a half. HIV vaccine is slightly more complicated, but it generally takes like 6 months to a year to make that cell line, and you could select the right clone, and that secretes the right kind of protein, because it has to go through many layers of like quality control. So now with mRNA, you kind of completely cut that 6 month to a year period. Now you could basically inject mRNA. You basically have to synthesize the mRNA lipid nanoparticle, which is basically synthesizing a primer or like a strand of DNA or RNA, which doesn't take long. You inject it, your body makes the protein, and you're good to go, like within no time. So in terms of if you're concerned about safety and other things, I don't think there was anything that was compromised. There were phase 1, phase 2, phase 3 trials done in the same way as you would do with a protein vaccine. And, all the precautions were taken. Obviously, there was some acceleration because multiple things were happening simultaneously. Like people invested because many companies got involved, and they invested into multiple phases of trials simultaneously. That kind of jump started, like each aspect of those. You have to test for efficacy, you have to test for safety, and other profiles. So in terms of the response, I think we were lucky that we had this mRNA technology, which kind of helped to respond to this pandemic quickly. And then the other part of your question was why we don't have a vaccine for HIV. Is that correct?
Yes, sir.
Yeah, so HIV is like a slightly more difficult, not slightly, I think it's a much more difficult surface to tackle. Like, in humans, like a lot of people make, literally everyone makes neutralizing antibodies to SARS-CoV-2 spike. And many people make broadly neutralizing antibodies. And, now we have more infected, vaccinated individuals. And we see that you can generate cross-neutralizing antibodies. And it's not the case with HIV. Even with natural infection, like if people get infected with HIV, 1 to 5% people make broadly neutralizing antibodies, which is kind of, which is not ideal. Like with a vaccine, you want to be more than at least 50% or more than 60% people should make those types of antibodies. And it particularly boils down to the complexity of the surface protein. It has a dense glycan shield. It has many other, it almost, the evasion strategies that I mentioned, it almost applies all of them. So for certain pathogens, for SARS, it's like variation, and probably immunodominance might be at some point once we are like, 4-5 years down the line in the pandemic, that might start, immunodominance might be a problem. But HIV literally, it applies all those evasion strategies to, evade your antibody-mediated immunity. So that's why it's very difficult to generate kind of effective antibodies with HIV vaccines.
Man, it
So
looks
thank
like
you so much.
like a sci-fi movie. Like the world is under the threat. You are on a mission critical, and you are one of the lead actors in there. How does it work when the pandemic started? What was your lab like, and how it is still maybe in the same situation?
So initially, when the pandemic hit, I think it was March 2020 when we started, when the cases started going up in United States, and I think mid-March we were told that we'll have to shut down, and only people who are working on coronaviruses will be allowed to work, and only half of their labs. So they have to pick people who can work as a part of this program. I think it was called something. I don't exactly remember the term, but it was like, yeah, something like that. What you do in a crisis mode. So my lab was not particularly working on coronavirus until then, and but we have been working on HIV on similar concepts and similar ideas, similar vaccine. So then we started kind of investing into coronaviruses, and then things kind of started picking up quickly. And basically we applied the same immunological or vaccine principles that we had learned for HIV to coronavirus, and kind of like there are common themes, and if you work on the right themes, obviously you'll get the right results. And in our case, I think we were lucky that we got some interesting results that we are continuing to pursue now for this virus, and potentially to viruses in future.
Thank you.
So due to the limitation of time, we have to wind up now. So I would once again like to thank you for joining us today. I really appreciate for your time today, and I would like to hand over it to Dr. Khalid.
Thank you so much, Umayya. Thank you so much, Dr. Said and Dr. Raiees Andrabi. Thank you so much. Before we formally wind it up, let's a little away from moving away from academia, that we do only once or twice a month. So, Dr. Raiees, what message do you have for people back home vis-a-vis number one, COVID? Is it for real? Number two, vaccines, and number third, vaccine hesitancy.
Yeah, well, I think India has done tremendously in terms of responding to SARS-CoV-2 pandemic. In terms of vaccine production and mass production, India is kind of famous because of the Serum Institute that had invested a lot into making vaccines, and it paid off eventually when it mattered the most. So I would say, like, initially, obviously the response was kind of late, and that led to some of the early waves that killed a lot of people. But after that, I think India has actually, given the population size, has remarkably responded to in terms of vaccination programs. And my suggestion to everyone, and hopefully, if you get a chance, get vaccinated. If you haven't gotten already vaccinated and are eligible, just take the vaccine, even if it doesn't prevent infection at some point. it will still reduce the severity of the disease. And hesitancy, it's not unique to one place. There's like definitely like vaccine hesitancy, and sometimes there are genuine concerns. And I think a lot of it depends on like how the messaging is spread, like, you know, how people are educated about vaccinations and vaccination programs, and how, you know, just doing every bit at your level, you know, educating people how vaccines work and why they are not harmful, and, you know, kind of debunking those myths. And vaccines are safe, you know. You take every group of human adults, and, you know, even pregnant women, or like any group, whoever is eligible, my strong suggestion would be get vaccinated, because it's the only way that you can prevent from getting infected or having a severe disease, unless we have some treatment options, which you know, companies are working on, and you know, it will take some time to get those available for general public. So yeah, I think that, yeah, that's it.
Thank you so much, Dr. Raiees. To add up, yeah, I agree with you. Creating awareness among masses and informing them the do's and don'ts about COVID-19 is going to help us for a long period of time. When this COVID started, I was on ground zero, a few kilometers away from Wuhan. I was in Shanghai, so I understood how they tackled it for a long. I mean, I believe that they did it very well. They handled it very well, and they disseminated the information that we have to do as well. And at JK Scientists, we have been doing it since day one. And we also thank you for sending that beautiful video. And I would inform you today that that video on YouTube has the highest number of hits we have on JK Scientists. Thank you so much. With this, we are really sorry. We have got so many questions that we are not able to take up. This session has been one of the hit sessions on our outreach programs. I hope that we can have more sessions in future with you, Dr. Raiees. Thank you once again for joining us. With this, I would like to take this opportunity to thank my teammates, Dr. Zahida Kamri, Dr. Saima Baig, Dr. Mir Zahur Gul, Umaya Muzaffar, and Dr. Asif for beautifully coordinating this event. And special thanks to Dr. Said Mubarak for joining us so early morning, and Umaya Muzaffar for joining us very late in Malaysia. Thank you so much, Dr. Raiees. Thank you so much, Dr. Said, and thank you, Umaya. Thank you so much.
Well, thank you so much for organizing this event and for this opportunity. And it was a real pleasure interacting with you all. And thanks for all the work that you guys are doing.
Thank you so much.
Thank you. Keep inspiring and give us vaccines. Khuda Hafiz.
Okay.
Thank you.
Bye-bye.
Take a clap for you.